Hello, and welcome to part 2 of chapter 15. So up to this point, we have been discussing the puzzle pieces that go together or go into the activation of your third line of defense. We are going to then apply those puzzle pieces to the activation process itself, starting with the activation of T helper cells. The activation of T helper cells is going to begin when an antigen is processed and presented to the T helper cell. The antigen processing and presentation must be done by an APC. Typically, APCs that are going to activate T helper include dendritic cells. Dendritic cells are going to be a type of phagocytic cell typically derived from a macrophage. What you're going to find is that dendritic cell is going to ingest and process the antigen. It is then going to make it more immunogenic and recognizable. What it is actually going to do, it is going to keep an epitope or the antigenic determinant of the antigen. It is then going to place that antigenic determinant on an MHC receptor, specifically MHC2, and it is going to move it to the surface of the APC. Once the MHC is presenting the antigenic determinant or epitope, it will then search for a T helper cell, which then can recognize it. T helper cells do not all recognize every epitope or antigenic determinant. There will be a very specific T helper cell that recognizes that epitope or antigenic determinant. Once that T helper cell comes in contact with that MHC2 receptor and the antigenic determinant that it recognizes, it will then bind to the APC. The APC and the T helper cell will basically form kind of a handshake where they will confirm self and confirm non-self or antigen. At that point in time, once confirmation is done, the APC will then secrete interleukin-1 onto the T helper cell. The T helper cell will then become activated. After the activation of the T helper cell, it will then secrete interleukin-2 in order to produce or recruit B cells and other types of T cells. So other T helpers as well as T cytotoxic cells can be activated by this T helper now. Typically, this process is going to be utilizing a T helper type 2 cell. This is the process in which we just discussed. Usually looking at a picture of the process is going to actually help understand the process. At the top of this picture, labeled number 1, you are looking at a dendritic cell. The dendritic cell has undergone phagocytosis of a foreign microorganism, otherwise known as an antigen. It has processed that antigen, and it has kept the antigenic determinant or epitope and made it more immunogenic and recognizable to T helper. It then will display it out on its appendages on the outside of its surface. You will see it's displayed within an MHC2 receptor. In the initial picture, number 1 and 2, you will see there are multiple MHCs with antigenic determinants within them. Only one of those antigenic determinants will be recognizable by one T helper. Multiple antigens can be displayed on a dendritic cell at one time. Once we find a T helper cell that recognizes one of these antigenic determinants, it will then communicate through that MHC2 receptor. When you see the dendritic cell and the T helper cell linked up in this location in the middle of the slide, what you are seeing here is what I referred to as the handshake. In the inset picture labeled number 3, you can see an up-close version of this handshake. What is occurring here is the APC is actually utilizing its MHC2 to have a chemical communication with T helper cell. MHC2 is displaying the antigenic determinant to the T helper cell. The T cell receptor is receiving the message of the antigenic determinant or epitope. It is confirming that antigen as non-self. At the same time, you can see CD4 has a physical connection to MHC2. What is happening here is CD4 is communicating with MHC2, and it is confirming self as well as non-self. At this point of confirmation of self and non-self entities, APC will then secrete interleukin-1 onto the T helper cell, activating it. The activation of T helper will then allow for that T helper to release other interleukins, which will assist in the activation and recruitment of other T cells and B cells. One of the things that this interleukin secretion will do is actually draw B cells to the T helper cell, which have also recognized the same antigen. Here in this slide, you will actually see at the top the same thing that we just discussed with the activation of a T helper cell by a APC. As I mentioned, most of the time when we're talking about activation of third line of defense, we are dealing with a T helper 2. The T helper 2 is basically what is going to activate a B cell. We'll get into the activation of the B cell in just a moment. As part of the activation process of a T helper cell, you will also see that it differentiates into memory T cells. Memory T cells are what give us the ability to recall antigen, and then therefore will not need to be reactivated upon secondary infection with the same antigen. You can also see that T helper 1s can also come from the activation process. T helper 1s are going to have a major role in allergic reactions as well as tissue rejection during transplantation. Another thing to go over is going to be the bottom half of this slide. In the bottom half of this slide, you can see the activation of a cytotoxic T cell. We know it's a cytotoxic T cell due to the fact that it's labeled with a CD8 marker. CD8 and cytotoxic T are synonymous. The activation process is very similar to the T helper. The main difference is that we do use an MHC class 1 versus the MHC class 2 that is seen with the T helper cell. So there is a form of a handshake. The MHC class 1 will display the antigenic determinant to the CD8 cell. The CD8 itself will link to the MHC1, confirming self, as well as the TCR or the T cell receptor will receive the antigen and confirm non-self. Upon the confirmation of self and non-self, we will have activation of your cytotoxic T cell. Once a T cell is activated that is cytotoxic, it will actually hunt and destroy cells. A lot of the times we find that cytotoxic T cells are active against viral infections. Viruses, being a type of obligate intracellular parasite, will live inside of our cells. The cytotoxic T cell will seek out those cells which are infected with virus and destroy them. Upon destroying our own body cells infected with virus, you are stopping the virus from reaching maturity. Maturity basically means that it cannot spread. Unfortunately, it will also damage or lyse our own body cells. Next up, let's talk about the activation of a B cell. First of all, B cell maturation does occur when it comes into contact with its first antigen. So a B cell is basically going to be produced by your bone marrow. After the production of a B lymphocyte by the bone marrow, it will then go to specific lymph nodes, spleens, or other associated lymphatic tissues, and it'll wait to come into contact with antigen. Once it comes into contact with antigen, it is now mature. Once a B cell is mature, it will then be activated by an interaction with a T helper cell. B cell activation then leads to antibody production. Once a B cell processes an antigen, it will need to interact with a T helper cell that has actually been activated against the same specific antigen. Once they have interacted, it will stimulate the B cell to undergo differentiation and clonal expansion. This will then give rise to plasma cells and memory cells. Plasma cells are the type of cell that is responsible for antibody production. Memory cells will allow for those B cells to not have to undergo activation again, and then will be automatically active to the antigen again later. This is the activation process to B cells. At the top of the slide, you can see labeled with number 1, we have a B cell interacting with an antigen. Just like the T cells, B cells are going to be selective to what kind of antigen they can bind to. All of the different Y-shaped B cell receptors that are found on the top of a B cell will vary from one to the next, meaning that they will be able to interact with various amounts of antigen. Whenever an antigen interacts with a B cell receptor that is specific for that antigen, it will then mature it. After maturation, the B cell will then display the antigenic determinant on its MHC2 receptor. At that point, it must seek out and find a T helper cell that has been activated against the same antigen. Once it has been activated against the same antigen, what you will find is another handshake. The T helper cell and the B cell will then shake hands. As you can see, the B cell is acting very much like the antigen presenting cell did during the T helper cell activation process. The handshake is very similar to that as well. And this is why we call B cells an antigen presenting cell. They are only going to present the antigen to a T helper cell after the T cell is activated. A B cell is not going to activate a T helper. Once they have a handshake, we again see MHC2 displaying the antigenic determinant. The T helper cell receives the antigen in its T cell receptor, and it interacts the CD4 and the MHC2. The conversation that they are having there is confirming self through the CD4 and MHC2, and confirming non-self between MHC2 and the TCR. T helper is basically confirming, yes, Mr. B cell, this is the antigen. Now let's activate you. T helper cell will then secrete a group of interleukins and B cell growth factor. The T helper cell secretes these chemicals onto the B cell. At this point, the B cell is activated. At this point, the B cell will then undergo division and clonal expansion. What this means is that it will divide in half first, and then those cells that arise from that division will then expand their numbers. Half of the activated B cell will become memory B cell. The other half of the activated B cell will become plasma cell. What we will find is memory B cells will then be able to remember the specific antigen and will not have to undergo interaction with the T helper to become activated again. They are already pre-active. Plasma cells are going to produce antibodies. During their first activation, they do produce IgM antibodies. After the very first IgM antibody production, those plasma cells will then class switch and they will start producing long-term immunity antibodies known as IgG. Plasma cells do not live as long as memory B cells. Plasma cells have a short cell life, whereas memory B cells have a longer cell life. A recap of the flow of the third line of activation. In order to get specific antibody production, you must first start off with an antigen-presenting cell eating a pathogen, keeping that antigenic determinant, otherwise known as epitope. It will then have to display the antigenic determinant on the MHC2, showing it to a T helper cell. The APC can then activate the T helper after the secretion of interleukin-1. That same T helper then seeks out a mature but inactive B cell that has found the same antigen. Once the T helper finds this B cell, it activates it by secreting B cell activation factors and a group of interleukin. Once B cell is active, it creates memory and antibody. Those memories and those antibodies are specific to the antigen which they were activated to. Looking into antibodies, we must understand their structure and their function. First of all, we've already talked about immunoglobulins as a form of B cell receptor. B cell receptors are antibodies. Therefore, an antibody structure is very much the same as a B cell receptor. It is made up of immunoglobulin, which is a large Y-shaped protein. It consists of 4 polypeptide chains. 2 of them are identical fragments known as the constant, and then 2 of them will be a variable. The Fab region is going to be the top part of the Y. The FC region is the bottom part of the Y. The FC region is basically where cells and molecules of the immune system are able to bind. Here is a picture of your immunoglobulin or antibody. As you can see in picture A, we have the Fab region labeled as the top Y shape or V shape. You can see the very tips of that Fab region include the variable region. The variable region contains within it the antigen binding sites. Once you have produced an immunoglobulin or antibody that is specific to an antigen, these variable regions will not change. They are only variable because they're different between the type of antibody produced for one antigen to the next. At the bottom of the Y-shaped structure, you will find the FC fragment. The FC fragment is basically the stick of the Y. The very end of it, you can see binding sites for cells occur here. This is basically where other immunologically active cells can bind to the antibody in order to carry out functions of destruction. You can also see the little notch that is found in the side of the FC fragment is labeled as the complement binding site. The complement binding site is also known as the complement fixation site. This is going to be a site at which complement can be stimulated for the classical pathway. As far as the functions of an antibody are concerned, one common misconception is that antibody destroys antigen. It actually does not. What antibody does is it actually aids the rest of the immune system in destroying antigen that it didn't recognize in the first place. One of the ways that antibodies actually are going to help destroy things is through indirect mechanisms, such as opsonization. Opsonization is a process of coating microorganisms or other particles with specific antibodies so that they are more readily recognized by your phagocytes. It basically is a coating of the cell surface of a bacteria cell or a coating of an antigen of other sorts. This then allows for a phagocyte to see it and eat it or undergo phagocytosis more readily. Another part of the antibody interaction that can occur is agglutination. Agglutination is basically where you have one variable region attached to one type of, or one antigen, and the other variable region is going to be attached to a completely other antigen, of course, of the same variety. This will allow for cross-linking of cells and forming large clumps of antigens and antibodies. You can also find that antibodies are useful for neutralization. Antibodies can fill the surface receptors on a virus or the active site on a microbial enzyme to prevent it from attaching. So in other words, it inhibits the virus or the microbial enzyme and therefore will not allow it to harm us. Some specific examples of neutralization would be antitoxins that are given for neutralizing bacterial exotoxins, or plainly, you can look at the Tdap and DTaP vaccinations. Tetanus and diphtheria are toxoid vaccines, which means that they are vaccines that stimulate antibody production against the main toxins associated with those diseases. So what we find is that those diseases cannot harm us due to the fact that we are blocking the active site of those microbial enzymes with our own antibodies. These are pictures of some of the things I just described. Starting with the picture in the upper left-hand corner, this is part of opsonization. Opsonization is where the antibody coats the full surface of the bacterial cell here, making it more recognizable by our macrophage. Our macrophage then can come by and engulf them through phagocytosis. Next up, we have neutralization. Neutralization is where the antibodies block the binding sites of a virus. The binding sites of a virus are basically the little spikes that stick out of the viral surface. These are necessary for attaching to a human host cell. When an antibody blocks them, it allows for the virus to not attach and therefore not cause disease. Now on the bottom, we have agglutination. Agglutination is going to be a cross-linking of cells. As you can see, it's where an antibody attaches to two different antigens of the same variety at the same time. Next, we have the complement fixation. Whenever we have antibody bound to antigen, it can stimulate the classical pathway of the complement system. What you are seeing here is just that. The complement fixation region on the Fc fragment of the antibody is recognized by the first protein of the complement system. It then allows for the cascading of those 23 different proteins, forming holes in the surface of the bacterial cell. Once we form enough holes in the surface of the bacterial cell, it begins to leak and basically loses all of its innards, including its DNA. Last but not least, we have precipitation. Basically, precipitation is very similar to agglutination. Instead of larger cells, we basically see with precipitation in smaller molecules. Now, when it comes to antibodies, antibodies do come in different varieties. All antibodies were not created equal. We do have 5 different classes of antibody or immunoglobulin, and they are going to spell MADGE, M-A-D-G-E. So IgM, IgA, IgD, IgG, and IgE. The IG simply stands for immunoglobulin. And so if I'm saying IgM, what that means is immunoglobulin type M. So IgM is basically known as your first responder. The first responder antibody is what is produced during the first activation of the B cell. You can also find low levels of IgM produced by B cells that are only activated, that are only mature and not activated. This is very minor and is not part of our traditional third line of defense activation. IgA is a secretory antibody. What this means is that it's basically an antibody that is favored in mucous membranes. You do find that IgA is going to be the type of antibody that a mother would share with her child during breastfeeding. IgD is a B cell receptor. As I previously mentioned, IgD, otherwise known as a B cell receptor, is going to be affixed to B cells. IgD does not free float in blood or plasma. IgG is known as your long-term antibody. There should be the majority of your antibodies represented as IgG in your bloodstream. Last but not least, we have IgE. IgE is going to be a type of antibody that is produced during allergic reactions. Typically, they are stimulated for production by T helper cell type 1. And you are also going to see that they are present during worm infections. Worm infections are going to be seen very similarly to allergies, so the same pathway of activation and production of the IgE antibody is going to occur here. When I mention something like class switching, what I'm talking about is that a B cell will stop producing one type of immunoglobulin and start producing another type of immunoglobulin. A lot of the times we talk about class switching between IgM and IgG, but it can also be from IgE to IgG. This is a function that we'll talk about in chapter 16. When it comes to primary and secondary responses to antigen, as you can realize, the primary response is going to be a slower mechanism. You are eventually going to produce your antibodies here, but typically the antigen that is overwhelming your body will cause illness upon that first exposure. The hope is that you actually produce enough memory B cell during that exposure and IgG that when you have a secondary exposure to that same antigen, that you have a more rapid response by your third line of defense. Due to this rapid response, typically you will not get sick. You will also have a stronger response due to the memory cells, so typically you do not see illness with that secondary exposure. This is a figure from your book, 15.3, that actually shows you the shape and structure of your immunoglobulins. As you can see, a lot of the immunoglobulins take on different varieties of shapes, as well as different functions. So IgG is going to be your main long-term memory antibody. It's also going to be the type of antibodies we produce whenever we are vaccinated. IgA is going to be your secretory antibody, typically favored to be found in mucous membranes, also the antibody that will be secreted from a breastfeeding mother to child. IgM is your first responder antibody. IgD is your B cell receptor. It is your main B cell receptor. Occasionally an IgM will serve as a B cell receptor, but IgD is the main one. IgE is going to be your antibody that is found in allergic reactions and your worm infections. What you see here in the form of IgM specifically is that it is a pentamer. The pentamer form here basically means that there are 10 different binding sites to antigen at one time. So this being said, you can actually see what the rationale is for producing it as your first antibody when you have your first initial activation of your B cell. Now what we do find is that IgM is basically like a rough draft. The antigen binding sites are not as good as they are in an IgG. An IgG is going to basically be a tight-fitting antigen binding site so that when it actually attaches to an antigen, it holds on tight, it won't let go. Whereas the antigen binding sites found in IgM, even though they're plentiful, they are not as tight-fitting, meaning that they will actually loosely fit to the antigen instead of tightly fit to the antigen. So this is why we don't produce IgM all the time. Instead, we produce IgG. Now that we have gone through the activation process associated with your third line of defense, we should also classify your specific and acquired immunities, which actually are coming from your third line of defense. So to introduce you to a few terms, we'll then put these terms together to describe different forms of immunity. First of all, you can have active immunity. Active immunity basically creates memory. Active immunity takes time and is long-lasting. When you have an active memory, it typically means that you have activated T helper, activated B cells, and you have formed an antibody and a memory cell. When you have a passive immunity, this does not activate your third line of defense. You're basically borrowing preformed antibodies from an individual that will give you a short-term or short-lived protection. Natural immunity is something that is acquired as part of normal life experiences, whereas artificial immunity is acquired through a medical procedure. So now we can put these four terms together and classify specific types of immunity. For example, you can have natural active immunity. Just looking at the terms natural and active here, natural means it's going to be a natural life experience, and active means it's going to be a long-term effect. Natural active immunity is when you acquire immunity upon infection and recovery. Natural passive immunity is going to be when you are a child and you acquire antibodies from your mother through the placenta, which will be IgG, and breast milk, which will be IgA. So natural experience, but only a temporary antibody presence. You do not make memory from this. Artificial active immunity, this is basically a vaccination. So you're artificially being stimulated to make an active immune system. Artificial passive immunity, this is going to be the same concept as natural passive, but it's going to actually be done through scientific or artificial means. Basically, we harvest antibodies and we are going to administer them to a patient through usually an IV drip bag. Where would we use artificial passive immunity? Well, there's actually a couple of different places we do use this type of therapy. So if you are an immunocompromised patient, you can actually use this for replacing antibodies for a short-term protection. One such instance would be somebody who is undergoing either cancer treatments or has had their bone marrow wiped out waiting for a bone marrow transplant. Immunotherapies can actually protect that patient in the short term until they get their bone marrow transplant or until they recover from their cancer treatments. You can also find that artificial passive immunity is utilized in the treatment of diphtheria, botulism, and snake and spider bites. So basically, the artificial passive immunity is going to neutralize the toxins associated with these diseases or bites. These are going to be immediate actions, but they are only going to last up to 2 or 3 months. Now, that being said, if you get bitten by the same snake twice in less than 3 months, it is recommended to get the artificial passive immunity if it has been even within the 2 to 3 months. Artificial active immunities are vaccinations. So vaccinations basically are going to be something that are recommended for all healthcare workers. You need to have all of your vaccines prior to going to work. And of course, they're recommended for children as they become school-aged and then through their schooling years. So this table actually shows us the schedule of vaccinations or recommended immunizations for people under the age of 18, basically showing us what they're supposed to have through their school-aged years. A lot of vaccinations are going to take place before they're really 2 or 3 years old. Their beginning vaccinations are going to include things like hepatitis B, rotavirus, diphtheria, pertussis, and tetanus, the Hib vaccine, pneumococcal vaccine, polio vaccine, flu vaccine, MMR vaccine, chickenpox vaccine, and hepatitis vaccine. Some vaccinations are only recommended for those people who are in high risk groups. So for example, the meningococcal vaccine is only going to be recommended for those people who are high risk. So a senior in high school going off to a college and living in a dormitory would be a perfect example of that. You can also see there are other vaccines that are recommended for other age groups, such as the HPV vaccine. This is recommended for children who are somewhere between the age of 8 and 10 and above. Definitely not for younger children. And then your other vaccines include a follow-up Tdap for a child that is above the age of 7. So these are all of your recommended vaccines, and most of these are also going to be recommended for healthcare workers. So making sure that you have things like your Tdap up to date, your MMR up to date, your flu shot annually, that you have antibodies against varicella, which is chickenpox, as well as other types of vaccinations. So here we do have vaccinations that are licensed within the United States, but they're not commonly given to the United States individuals, such as tuberculosis and typhoid and anthrax. These 3 specifically are only going to be given for very high-risk occupations or military. Beyond that, meningitis and pneumococcal pneumonia are only going to be given to also high-risk individuals. When it comes to toxoid vaccines, that Tdap vaccine is going to be a good example, covering diphtheria and tetanus. Both of these are going to be vaccinations against the toxin. You also have what we call inactivated whole virus vaccines. Inactivated whole virus vaccines basically means that we have a virus that's been killed. So it's not a living virus. And it is given to the patient in order to promote an immune response without actual disease. Those that contain live attenuated viruses, these are basically living viruses that have been weakened. So some of the more common ones would be the varicella vaccine. You also have an MMR vaccine represented here, which is measles, mumps, and rubella. Some subunit viral vaccines are basically going to be vaccines that are only against the antigenic determinant or epitope. And then we do have some examples of combined vaccines, which are very popular. The Tdap and the MMR are very popular in the United States. But you also can find that some of the meningitis vaccines are combination vaccines as well. This ends part 2 of chapter 15.